Hello YouTube, my name is Neil and we're studying for the MCATs today. We're in the Biochemistry Review, Chapter 9, Carbohydrate Metabolism 1, Glycolysis, Glycogen, Gluconeogenesis, and the Pentose Phosphate Pathway. Maintaining a constant blood glucose concentration around 100 milligrams per deciliter, or 5.6 millimolar, is of the utmost importance in the body. High blood sugar causes long-term damage to the retina, kidney, blood vessels, and nerves, while low blood sugar can cause autonomic disturbances, seizures, and even coma. Without the ability to take in glucose constantly, the body must find ways to store and release glucose as it is needed. And given the variety of food we eat on a daily basis, the body must find ways to use all of the various carbohydrates it takes in. There's a complex interplay between the neurological, endocrine, digestive, and excretory systems to maintain this blood glucose concentration, much of which is discussed in Chapter 12. In this chapter, we'll take a look at the metabolic pathways that involve glucose, the methods by which our bodies digest glucose and other monosaccharides, store and release glucose for energy, generate glucose from other biomolecules, and use glucose to create some of the coenzymes and substrates needed for biosynthesis. This chapter is the first of 4 that focuses on metabolism in the Biochemistry Review. Here we focus on metabolic processes of glucose and do not require oxygen. In Chapter 10, we'll turn our focus to the processes that only occur under aerobic conditions. In Chapter 11, we'll explore the metabolism of lipids and amino acids. And finally, in Chapter 12, we'll bring all of metabolism together with a focus on bioenergetics and the regulation of metabolism overall. Glucose transport. Glucose entry into most cells is driven by concentration and is independent of sodium, unlike absorption from the digestive tract. Normal glucose concentration in peripheral blood is 5.6 millimolar. Normal range is 4 to 6. There are 4 glucose transporters called GLUT1 through GLUT4. GLUT2 and 4 are the most significant of these because they're located only in specific cells and are highly regulated. GLUT GLUT2 is a low affinity transporter in hepatocytes and pancreatic cells. After a meal, blood traveling through the hepatic portal vein from the intestine is rich in glucose. GLUT2 captures the excess glucose primarily for storage. When the glucose concentration drops below the Km for the transporter, much of the remainder bypasses the liver and enters the peripheral circulation. The Km of GLUT is quite high, 15 millimolar. This means that the liver will pick up glucose in proportion to its concentration in the blood, first-order kinetics. In other words, the liver will pick up excess glucose and store it preferentially after a meal. When blood glucose levels are high, in other words, the liver will pick up excess glucose and store it preferentially after a meal when blood glucose levels are high. In the beta islet cells of the pancreas, GLUT2 along with the glycolytic enzyme glucokinase serves as the glucose sensor for insulin release. Alright, so GLUT2 sits in the hepatic portal vein and intestines. The Km is high, so usually it is allowing most of the glucose to go into the circulation. It's just when it's really high, it'll start storing it. So let's note that normal glucose, blood glucose, 4 to 6 millimolar. GLUT2, when glucose is around 15 millimolar. GLUT2 stores glucose. Along with glucokinase and GLUT serve as a glucose sensor for insulin release, so they trigger insulin. GLUT4 is in adipose tissue and muscle and responds to the glucose concentration in peripheral blood. The rate of glucose transport in these two tissues is increased by insulin, which stimulates the movement of additional GLUT4 transporters to the membrane by a mechanism involving exocytosis, as shown in figure 9.1. The Km of GLUT4 is close to the normal glucose levels in blood, around 5. This means that the transporter is saturated when blood glucose levels are just a bit higher than normal. When a person has high blood sugar concentrations, these transporters will still permit only a constant rate of glucose influx because they will be saturated, zero-order kinetics. Then how can cells with GLUT4 transporters increase their intake of glucose? By increasing the number of GLUT4 transporters on their surface. Although basal levels of transport occur in all cells independently of insulin, the transport rate increases in adipose tissue and muscle when insulin levels rise. Muscle stores excess glucose as glycogen, and adipose tissue requires glucose to form dihydroxyacetone phosphate, DHAP, which is converted to glycerol phosphate to store incoming fatty acids as triacylglycerols. All right, GLUT4 in adipose and muscle. So insulin causes increase in GLUT4 transporters. via exocytosis. The Km of glut let's say Km is around 5 millimolars. And I guess between muscle and adipose tissue, muscle stores glycogen, while adipose tissue requires glucose to form DHAP, dihydroxyacetone phosphate, which is converted to glycerol phosphate to store incoming fatty acids as triacylglycerols. Glycerol phosphate to triacylglycerol. All right, compare and contrast GLUT2 and GLUT4. GLUT2 is in the liver, or in the intestines and the hepatic portal vein, and GLUT4 is in fat and muscle. The Km of GLUT2 is high, like 15, while the Km of GLUT4 is like 5, around normal physiological blood levels. Saturated at normal glucose levels. GLUT2 is not, GLUT4 is. Responsive to insulin. I guess GLUT2 initiates insulin, GLUT4 responds to insulin. How does insulin promote glucose entry into the cell? By promoting more GLUT transporters to move to the membrane. Let's see those answers real quick. Important tissues, liver and pancreas. Okay. For GLUT4, adipose tissue and muscle. The Kms are 15 and 5. Saturated at normal levels, no, yes. Responsive to insulin, no, but serves as glucose sensor to cause release of insulin in pancreatic beta cells. And yes for GLUT4. So important tissues, liver. So hepatic portal vein is liver. And pancreas also. Let's add pancreas. Oh, and how insulin promotes glucose entry. GLUT4 is saturated when glucose levels are only slightly above 5, so glucose entry can only be increased by increasing the number of transporters. Insulin promotes the fusion of vesicles containing preformed GLUT4 with the cell membrane. Great. Glycolysis. All cells can carry out glycolysis. In a few tissues, most importantly red blood cells, glycolysis represents the only energy-yielding pathway available because red blood cells lack mitochondria, which are required for the citric acid cycle, electron transport chain, oxidative phosphorylation, and fatty acid metabolism. Glucose is the major monosaccharide that enters the pathway, but others such as galactose and fructose can also feed into it. Let's write down that all the citric acid cycle, the electron transport chain, oxidative phosphorylation, and fatty acid metabolism are all in the mitochondria. So this is not in the mitochondria. I thought this does still involve the mitochondria. Maybe just the last step. Oh, here we go. The pyruvate part is in the mitochondria. Galactose and fructose. Glucose, galactose, and fructose can all utilize glycolysis. Glycolysis is a cytoplasmic pathway that converts glucose into 2 pyruvate molecules, releasing a modest amount of energy captured in 2 substrate-level phosphorylations and 1 oxidation reaction. If a cell has mitochondria and oxygen, the energy carriers produced in glycolysis, NADH, can feed into the aerobic respiration pathway to generate energy for the cell. If either mitochondria or oxygen is lacking, such as in erythrocytes or exercising skeletal muscle, respectively, glycolysis may occur anaerobically, although some of the available energy is lost. Glycolysis also provides intermediates for other pathways. In the liver, glycolysis is part of the process by which excess glucose is converted to fatty acid for storage. So glucose to pyruvates and energy. So both aerobic and anaerobic. The aerobic and aerobic more efficient. All right. Because of NADH is used in other processes. Important enzymes of glycolysis. While glycolysis contains many different steps as illustrated in Figure 9.2, the MCAT predominantly tests on the enzymes that are highly regulated or that serve an important energetic function. Therefore, we'll focus our attention on 5 of these enzymes. So yeah, I guess we'll just write down the ones that they want us to focus on, because this is a lot to write down. But you can see glycolysis starts with glucose, gets turned into 6P glucose, 6-phosphate glucose, I guess. Fructose 6P, fructose let's see. So I guess we just have multiple alternate pathways. We have one kind of alternate path using insulin and PFK2 to make fructose 2,6 biphosphate, which then gets turned into and they don't say what turns this into fructose, but anyways, fructose 6 phosphate turns into fructose 1,6 biphosphate, and then that gets turned into glyceraldehyde 3 phosphate, and then we have this step with the NADH they were talking about. If you're doing aerobic respiration, I guess you would maybe take this. You'd use NADH instead. If you weren't using NADH, glyceraldehyde would be turned into 1,3 biphosphoglycerate, and then phosphoglycerate, 3 phosphoglycerate, and then 2 phosphoglycerate. So a lot of like reorganizing the same molecules, right, or the same atoms. Phosphoenol pyruvate, PEP. Finally to pyruvate. And then pyruvate is going to go into the mitochondria and do other things. Is this considered part of glycolysis? No, this is just other things. Fatty acid synthesis, the TCA cycle is the, what is that? Well, we'll learn in the next chapter. I guess that's the oxidative part. All right. The first enzymes we're going to talk about are hexokinase and glucokinase, which are the first step of glucose to glucose 6-phosphate. Hexokinase and glucokinase. The first steps in glucose metabolism in any cell are transport across the membrane and phosphorylation by kinase enzymes inside the cell to prevent glucose from leaving via the transporter. Remember from chapter 2 that kinases attach a phosphate group from ATP to their substrates. Glucose enters the cell by facilitated diffusion or active transport. In either case, these kinases convert glucose to glucose 6-phosphate because the GLUT transporter are specific for glucose, not phosphorylated glucose. The glucose gets trapped inside the cell and cannot leak out. Hexokinase is widely distributed in tissues and is inhibited by its product, glucose 6-phosphate. Glucokinase is found only in liver cells and pancreatic B islet cells. In the liver, glucokinase is induced by insulin. Table 9.1 identifies the differences between these enzymes. These coincide with the differences between the glucose transporters in these tissues. Some differences between hexokinase and glucokinase. Hexokinase is everywhere. Glucokinase is in the liver. Here it says that glucokinase is induced by insulin. Here it says along with GLUT2 acts as the glucose sensor. So it induces insulin. I don't know why they said it is induced by insulin. Oh, in the liver specifically, glucokinase is found in liver cells and pancreatic beta islet cells. In the liver, glucokinase is induced by insulin. either way, I guess, what do we write up here? Insulin. Oh, right. GLUT2 causes insulin. Insulin causes GLUT4. Right. Hexokinase has a low Km. Glucokinase has a high Km. Inhibited hexokinase is inhibited by glucose 6-phosphate, and glucokinase is induced by insulin in hepatocytes. So glucose to glucose 6-phosphate by a kinase is phosphorylated. Hexokinase is all cells, it's in all cells, has a low Km and inhibited by G6P, glucose 6-phosphate. Glucokinase in the liver has a high K sub M and induced by insulin. All right, next, phosphofructokinase, PFK1 and 2. PFK1 is the rate-limiting enzyme and main control point in glycolysis. In this reaction, fructose 6-phosphate is phosphorylated to fructose 1,6-biphosphate using ATP. PFK1 is inhibited by ATP and citrate and activated by AMP. This makes sense because the cell should turn off glycolysis when it has sufficient energy, high ATP, and turn on glycolysis when it needs energy, high AMP. Citrate is an intermediate of the citric acid cycle, so high levels of citrate also imply that the cell is producing sufficient energy. Fructose 6-phosphate, should I just write P, to fructose 1,6-biphosphate. How do they write it over here? 6-bis space P. One specifically, rate limiting step. It is inhibited by, or downregulated by ATP and citrate, and increased by AMP. Insulin stimulates and glucagon inhibits PFK1 in hepatocytes by an indirect mechanism involving PFK2 and fructose 2,6 biphosphate, as shown in figure 9.2. Insulin activates phosphofructokinase 2, which converts a tiny amount of fructose 6-phosphate to fructose 2,6 biphosphate. Fructose 2,6 biphosphate activates PFK1. On the other hand, glucagon inhibits PFK2, lowering fructose 2,6 biphosphate and thereby inhibiting PFK1. PFK2 is found mostly in the liver. By activating PFK1, it allows these cells to override the inhibition caused by ATP so that glycolysis can continue even when the cell is energetically satisfied. The metabolites of glycolysis can thus be fed into the production of glycogen, fatty acids, and other storage molecules rather than just being burned to produce ATP. Did they say PFK1 was throughout the body? Sure. PFK2 is found mostly in the liver. So in the liver, insulin activates PFK2, which takes F2 or F6P to F26. They just go BP instead of this BIS-P. Instead of 1,6, it's 2,6, which then goes and activates PFK1. And of course, glucagon does the opposite. All right. Next, glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase catalyzes an oxidation and addition of inorganic phosphate to its substrate, glyceraldehyde 3-phosphate. This results in the production of a high-energy intermediate, 1,3-biphosphoglycerate, and the reduction of NAD to NADH. If glycolysis is aerobic, the NADH can be oxidized by the mitochondrial electron transport chain, providing energy for ATP synthesis by oxidative phosphorylation. Great. Does oxidation and addition of inorganic phosphate, Pi, to G3P to make 1,3-biphosphoglycerate. And NAD plus to NADH. So you got your NADH there. Next, 3-phosphoglycerate kinase. Phosphoglycerate kinase transfers the high-energy phosphate from 1,3-biphosphoglycerate to ADP, forming ATP and 3-phosphoglycerate. This type of reaction, in which ADP is directly phosphorylated to ATP using a high-energy intermediate, is referred to as substrate-level phosphorylation. In contrast to oxidative phosphorylation in mitochondria, substrate-level phosphorylations are not dependent on oxygen and are the only means of ATP generation in an aerobic tissue. So we take 1,3-biphosphoglycerate to 3-phosphoglycerate, takes one of those phosphates, but also ADP gets turned into ATP. Substrate level phosphorylation, meaning not oxidative, meaning anaerobic. Next, pyruvate kinase. In the absence of oxygen, fermentation will occur. The key fermentation enzyme in mammalian cells is lactate dehydrogenase, which oxidizes NADH to NAD+, replenishing the oxidized coenzyme for glyceraldehyde 3-phosphate dehydrogenase. Without mitochondria and oxygen, glycolysis would stop when all the available NAD+ had been reduced to NADH. By reducing pyruvate to lactate and oxidizing NADH to NAD+, lactate dehydrogenase prevents this potential problem from developing. There's no net loss of carbon in this process. Pyruvate and lactate are both 3-carbon molecules. In aerobic tissues, lactate does not normally form in significant amounts. However, when oxygenation is poor during strenuous exercise in skeletal muscle, a heart attack, or a stroke, most cellular ATP is generated by anaerobic glycolysis and lactate production increases. In yeast cells, fermentation is the conversion of pyruvate, 3 carbons, to ethanol, 2 carbons, and carbon dioxide, 1 carbon. While the end products are different, the result of both mammalian and yeast fermentation is the same, replenishing NAD+. All right. Oh, I forgot about pyruvate kinase. I just went right on to fermentation. Let's backtrack to pyruvate kinase. The last enzyme in aerobic glycolysis is catalyzes a substrate level phosphorylation of ADP using the high energy substrate phosphoenol pyruvate, PEP. Pyruvate kinase is activated by fructose 1,6 biphosphate from the PFK1 reaction. This is referred to as feed-forward activation, meaning that the product of an earlier reaction of glycolysis, fructose 1,6 biphosphate, stimulates or prepares a later reaction in glycolysis by activating pyruvate kinase. So pyruvate, what we'll do is F16BP what am I doing? So we use PEP, which is phosphoenol pyruvate, to something, but we also do that ADP to ATP. They don't tell us what the product is, do they? But we do know that it's activated by fructose 1,6 biphosphate from PFK1. Next, fermentation. Lactate dehydrogenase, which oxidizes NADH to NAD+. And it also takes pyruvate to lactate So, should we write down that in yeast pyruvate is turned into alcohol, ethanol. All right, important intermediates of glycolysis. Glycolysis serves as a crossroad for a number of metabolic processes. The intermediates of glycolysis are often used to link different pathways during both catabolism and anabolism. 3 of these intermediates are worth highlighting. Dihydroxyacetone phosphate, DHAP, is used in hepatic and adipose tissue for triacylglycerol synthesis. DHAP is formed from fructose 1,6 biphosphate. It can be isomerized to glycerol 3-phosphate, which can then be converted to glycerol, the backbone of triacylglycerols. DHAP is turned into glycerol phosphate 3-phosphate, which can be converted to glycerol, the backbone of triacylglycerols. Let's write that down a little more detailed. Intermediates include DHAP. Let's just start with fructose 1,6 biphosphate becomes G3P, which becomes glycerol. Triacylglycerol backbone. All right. 1,3-biphosphoglycerate and phosphoenol pyruvate, PEP, are high-energy intermediates used to generate ATP by substrate-level phosphorylation. This is the only ATP gained in anaerobic respiration. Phosphoglycerate, which I'm just going to write down as BPG, because they do, and phosphoenol pyruvate is PEP, and they're both substrate level phosphorylation, which means they're not oxidative, they don't require oxygen. Irreversible enzymes. Are these the only 2 intermediates? 3 intermediates in total. Irreversible enzymes. 3 enzymes in the pathway catalyze reactions that are irreversible. This keeps the pathway moving in only one direction. However, the liver must be able to generate new glucose from other biomolecules through gluconeogenesis, which is essentially the reverse of glycolysis. Because of the irreversible enzymes of glycolysis, different reactions and therefore different enzymes must be used at these 3 points: glucokinase or hexokinase, PFK1, and pyruvate kinase. So the irreversible ones are glucokinase slash hexokinase, PFK1, and pyruvate kinase. Glycolysis in erythrocytes. In erythrocytes, red blood cells, anaerobic glycolysis represents the only pathway for ATP production, yielding a net 2 ATP per glucose. Red blood cells have biphosphoglycerate mutase, which produces 2,3-biphosphoglycerate, 2,3-BPG, from 1,3-BPG in glycolysis. Remember that mutases are enzymes that move a functional group from one place in a molecule to another. In this case, the phosphate is moved from the 1 position to the 2 position. 2,3-BPG binds allosterically to beta chains of hemoglobin, A, and decreases its affinity for oxygen. This effect of 2,3-BPG is seen in the oxygen dissociation curve for HbA, shown in figure 9.3. The rightward shift in the curve is sufficient to allow unloading of oxygen in tissues, but still allows 100% saturation in the lungs. An abnormal increase in erythrocyte 2,3-BPG might shift the curve far enough so that HbA is not fully saturated in the lungs. So the saturation curve is affected by an intermediate in glycolysis. So if there's sugar, it'll unload more or it'll unload less. They have biphosphoglycerate mutase, which is going to take 2,3-BPG it's going to make 2,3-BPG from 1,3-BPG. Take 1,3-BPG to 2,3-BPG. Which increases hemoglobin A which is HbA. Increases HbA's affinity for oxygen. So it won't let go of oxygen as it usually would. It says the right shift in the curve is sufficient to allow unloading of oxygen in tissues, but still allows 100% saturation in the lungs. Decreases its affinity. Sorry, not increases, decreases. That makes more sense. Decreases affinity, so it unloads easily. Great. Although 2,3-BPG binds to hemoglobin A, it does not bind well to fetal hemoglobin, with the result that fetal hemoglobin has a higher affinity for oxygen than maternal hemoglobin. This allows transplacental passage of oxygen from mother to fetus. Fetal hemoglobin, or HbF, does not respond to 2,3-BPG. All right, what are the function and key regulators of the following enzymes? Which ones are reversible? Hexokinase turns glucose into glucose 6-phosphate. It's inhibited by G6P and induced by insulin in the liver, which is the glucokinase version. Never mind. So hexokinase is inhibited by G6P. Is it reversible? It is one of the non-reversible steps, if I remember it correctly. Glucokinase is similar, but instead of being inhibited by G6P, it's induced by insulin. Is it also inhibited by G6P? We'll see. It does the same thing, glucose to glucose 6-phosphate. It is also irreversible, I imagine. PFK turns fructose 6-phosphate into fructose 1,6-biphosphate. It is regulated by ATP, citrate, and AMP. It is not reversible. Glyceraldehyde 3-Phosphodehydrogenase turns G3P to 1,3-biphosphoglycerate. It is reversible. Pyruvate kinase turns phosphoenol pyruvate into pyruvate, I guess. Is that the last step? Phosphoenol pyruvate into pyruvate. It is regulated, it is activated by fructose 1,6-biphosphate. So that's the feed forward. Let's write down feed forward, right? Or is that what they called it? feed-forward activation. Is it reversible? No, it is also not reversible. Why must pyruvate undergo fermentation for glycolysis to continue? That's assuming that there's no oxygen. I guess that's in the case that there's no oxygen. Why is it necessary that fetal hemoglobin does not bind to 3-BPG? So that the fetal blood oxygen doesn't go too low. Because if it did bind, then it would probably, the oxygen wouldn't get to the baby from the mother. Like after a meal or something, and there's a lot of sugar in the blood. Let's see what the answers are for those. Number 1, hexokinase phosphorylates glucose to form G6P, trapping glucose in the cell. It's inhibited by G6P. It is irreversible. Glucokinase also phosphorylates glucose. It's induced by insulin. It is irreversible. PFK1 catalyzes the rate-limiting step of glycolysis, phosphorylating fructose 6-phosphate to fructose 1,6 biphosphate. It's inhibited by ATP, citrate, and glucagon. It's activated by AMP, fructose 2,6 biphosphate, and insulin. It is irreversible. So let's write some of those down. We didn't have all those ATP, citrate, and AMP, but also glucagon, activated by AMP, fructose 2,6-BP, and insulin. All right, it is irreversible. Glyceraldehyde 3-phosphate dehydrogenase generates NADH while phosphorylating glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate. It is reversible. 3-phosphoglycerate kinase performs a substrate-level phosphorylation, transferring a phosphate from 1,3-biphosphoglycerate to ADP, forming ATP and 3-phosphoglycerate. It is reversible. I might have skipped one there. Pyruvate kinase performs another substrate-level phosphorylation, transferring a phosphate from phosphoenol pyruvate to ADP, forming ATP and pyruvate. It is activated by fructose 1,6 biphosphate, and it is irreversible. No, you didn't forget anything. Oh, wait. Maybe I did. Maybe I forgot this one. Maybe I skipped glyceraldehyde 3-phosphate dehydrogenase. No, I wouldn't have done. Maybe it was a... Anyways, number 2, fermentation must occur to regenerate NAD+, which is in limited supply in cells. Fermentation generates no ATP or energy carriers, merely regenerates the coenzymes needed in glycolysis. Okay, so fermentation is needed for NADH. Fermentation is necessary for NAD+ replenishment. Number 3, the binding of 2,3-BPG decreases hemoglobin's affinity for oxygen. Fetal hemoglobin must be able to steal oxygen from maternal hemoglobin at the placental interface. Therefore, it would be disadvantageous to lower its affinity for oxygen. Great. Before we continued, I wanted to find each of these enzymes in this greater pathway. We have the hexokinase and glucokinase right here in the beginning. Oh, they labeled them. 2 is going to be 1, 2, 3. I guess those are probably the rate limiting steps. So 2 is PFK1. We have PFK2 here, which stimulates PFK1, and PFK2 is stimulated by insulin. So that's what this plus means, I guess. Then the next one that we went over is down here, glyceraldehyde 3-P dehydrogenase. And this is a step that makes NADH out of NAD. So this is why we need more NAD plus from fermentation if oxygen is not present or mitochondria is not present. We have the 3-phosphoglycerate kinase. Is this one phosphoglycerate kinase? Takes 1,3-biphosphoglycerate to 3-phosphoglycerate. We have pyruvate kinase, which is our other rate limiting step, or our other irreversible step rather, which takes PEP to pyruvate. And fermentation regenerates your NAD+, and pyruvate goes on into the mitochondria. Other intermediates. So they have this alternative pathway for fructose 1,6-biphosphonate, which is the DHCP. Where did I write that down? I didn't write that down. But yeah, the DHCP to, yeah, I did. DHCP to G3P to glycerol, which is the backbone for triacylglycerols. All right. Other monosaccharides. While glucose represents the primary monosaccharide used by cells, other monosaccharides such as galactose and fructose can also contribute to ATP production by feeding into glycolysis or other metabolic processes. These monosaccharides are tested far less frequently than glucose on the MCAT, but are included here to compare and contrast their metabolism with glycolysis. In particular, notice the similarities between figure 9.2, glycolysis, and figures 9.4, galactose metabolism, and 9.5, fructose metabolism. First, we have galactose metabolism. An important source of galactose in the diet is the disaccharide lactose present in milk. Lactose is hydrolyzed to galactose and glucose by lactase, which is a brush border enzyme of the duodenum. Along with other monosaccharides, galactose reaches the liver through the hepatic portal vein. So first, let's just write down that lactose turns into galactose and glucose by lactase. Once transported into tissues, galactose is phosphorylated by galactokinase, trapping it in the cell. Galactokinase to make a galactose 1-phosphate. The resulting galactose 1-phosphate is converted to glucose 1-phosphate by galactose 1-phosphate uridyl transferase and an epimerase. Epimerases are enzymes that catalyze the conversion of one sugar epimer to another. Remember from chapter 4 that epimers are diastereomers that differ at exactly one chiral carbon. The pathway is shown in figure 9.4. Important enzymes to remember are galactokinase and galactose 1-phosphate uridyl transferase. So galactose 1P turns into glucose 1P by galactose 1P uridyl transferase and an epimerase. So I'm just going to make the enzymes blue. And And I take this out. I just don't like the way that looks. I'm going to put galactose plus glucose. We're only worried about the galactose in this case, so. So they say pay attention to the second two of those enzymes. Fructose metabolism. Fructose is found in honey and fruit and is part of the disaccharide sucrose, common table sugar. Sucrose is hydrolyzed by the duodenal brush border enzyme sucrase, and the resulting monosaccharides, glucose and fructose, are absorbed into the hepatic portal vein. The liver phosphorylates fructose using fructokinase to trap it in the cell. The resulting fructose 1-phosphate is then cleaved into glyceraldehyde and DHAP by aldolase B. Smaller amounts are metabolized in renal proximal tubules. The pathway is shown in figure 9.5. So in this case, we start with sucrose, which becomes fructose and glucose. So we got our fructokinase. Then we have a F1P. The resulting F1P is cleaved into glyceraldehyde and DHAP. And DHAP. So that is done by aldolase B, the fructokinase up here, and sucrase is the one that breaks the sucrose up into fructose and glucose. Great. Other tissues phosphorylate fructose slowly through hexokinase. But the liver and the kidney have fructokinase. This is write down that it's in the liver. And let's look back at this. This is also in the liver, isn't it? Lactase is in the intestines, but galactokinase is in the liver, brain, and other tissues. So I guess that's a little more common than the fructokinase, which is in the liver and kidney. in liver and kidney. All right. Should I also make a note that these are on the intestines. These are also in the gut. All right. Which enzyme is responsible for trapping galactose in the cell? What enzyme in galactose metabolism results in a product that can feed directly into glycolysis, linking the two pathways? Well, galactokinase traps galactose in the cell. And the part that could feed glycolysis, does the glucose 1-phosphate feed glycolysis? Yeah, I guess it turns into glucose 6P. And then it can enter glycolysis. Glucose 1P is not a part of glycolysis, is it? No, but glucose 6P is. So the linking enzyme, they don't tell us what the enzyme for this is, but I guess what gets you from galactose to glucose is epimerase. The uridyl transferase okay, so it's a uridyl transferase is the enzyme, is the linking enzyme to glycolysis, because that's how you get glucose from galactose. Trapping enzyme, galactokinase, uridyl transferase is the linking enzyme. Which enzyme is responsible for trapping fructose? That's fructokinase. And the enzyme that feeds into glycolysis, I guess, would be aldolase B. Because from aldolase B, you get glyceraldehyde, and from glyceraldehyde, you get glyceraldehyde 3-phosphate. Which is right there. Aldolase. Aldolase B, specifically. Aldolase is already used in glycolysis that takes fructose 1,6-biphosphate to glyceraldehyde 3-phosphate. Aldolase B will take fructose 1-phosphate to glyceraldehyde and eventually glyceraldehyde 3-phosphate. Great, let's check those answers. Galactose is phosphorylated to by galactokinase. Galactose 1P uridyl transferase produces glucose 1-phosphate, a glycolytic intermediate, thus linking the pathways. Fructose is phosphorylated by fructokinase, trapping it in the cell, and aldolase B produces DHAP and glyceraldehyde, which can be phosphorylated to form glyceraldehyde 3-phosphate, which are glycolytic intermediates, thus linking the pathways. Great. right. Pyruvate Dehydrogenase. Pyruvate from aerobic glycolysis enters mitochondria, where it may be converted to acetyl-CoA for entry into the citric acid cycle if ATP is needed, or for fatty acid synthesis if ATP is present. The pyruvate dehydrogenase complex, PDH, reaction shown in figure 9.6 is irreversible and cannot be used to convert acetyl-CoA to pyruvate or to glucose. Pyruvate dehydrogenase in the liver is activated by insulin, whereas in the nervous system the enzyme is not responsive to hormones. This makes sense because high insulin levels signal to the liver that the individual is in a well-fed state. Thus, the liver should not only burn glucose for energy, but shift the fatty acid equilibrium toward production and storage rather than oxidation. It says fatty acid synthesis discussed in chapter 11 starts from citrate produced in the citric acid cycle. So pyruvate to acetyl coenzyme A which can go to the citric acid cycle for ATP, or it can go to fatty acid synthesis. Pyruvate dehydrogenase complex, let's call it PDH, is irreversible. Activated by insulin in liver, non-reactive in other cells. So when you're well fed, high insulin levels, you store rather than burn. You got a chart of glucose turning into pyruvate due to glycolysis. And then with coenzyme A, pyruvate dehydrogenase will turn it into acetyl-CoA, which can then feed the citric acid cycle or fatty acid synthesis, as we've drawn here. Acetyl-CoA is an inhibitor of pyruvate dehydrogenase. So let's also write down that it's inhibited by acetyl-CoA. Whoops. All right. Pyruvate dehydrogenase is actually a complex of enzymes carrying out multiple reactions in succession. The details of each of these reactions are covered in Chapter 10, but an overview of the enzyme is provided here because it represents one of three possible fates of pyruvate: conversion of acetyl-CoA by PDH, conversion to lactate by lactate dehydrogenase, or conversion to oxaloacetate by pyruvate carboxylase. This large complex requires multiple cofactors and coenzymes, including thiamine, pyrophosphate, lipoic acid, coenzyme A, FAD, NAD+. Insufficient amounts of any of these cofactors or coenzymes can result in metabolic derangements. Pyruvate dehydrogenase is inhibited by its product acetyl-CoA. This control is important in several contexts and should be considered along with pyruvate carboxylase, the other mitochondrial enzyme that uses pyruvate. It's introduced in gluconeogenesis later in this chapter. Essentially, the buildup of acetyl-CoA, which happens during beta oxidation, causes a shift in metabolism. Pyruvate is no longer converted into acetyl-CoA to enter the citric acid cycle, but rather into oxaloacetate to enter gluconeogenesis. Did we already go over lactate dehydrogenase? Lactate dehydrogenase takes pyruvate to lactate. Let's write this down. Part of the PDH complex is pyruvate to lactate by lactate dehydrogenase and also conversion to oxaloacetate. Oxaloacetate by the enzyme Pyruvate Carboxylase. This large complex requires multiple cofactors and coenzymes, including thiamine, pyrophosphate, lipoic acid, CoA, FAD, and NAD. Let's write these down. Cofactors, coenzymes, include thiamine, pyrophosphate, lipoic acid, coenzyme A, FAD, NAD+. All right. So when acetyl-CoA production is inhibited, it seems that pyruvate carboxylase is actually preferred. Pyruvate carboxylase is preferred for oxaloacetate. Or gluconeogenesis. All right, what are the reactants of the pyruvate dehydrogenase complex, and what are the products? Reactants: pyruvate, products, acetyl-CoA, or lactate or oxaloacetate. How does acetyl-CoA affect PDH complex activity, and why? In high concentrations of acetyl-CoA, PDH will shift from production of acetyl-CoA to oxaloacetate for gluconeogenesis. The reactants are pyruvate, NAD+, and CoA are the reactants. Pyruvate plus NADH plus CoA. Acetyl-CoA, acetyl-CoA, NADH, and CO2 are the products. So acetyl-CoA plus NAD. All right, so not NADH over here, NAD plus over here, NADH, and CO2 are the products. Acetyl-CoA inhibits the PDH complex as a product of the enzyme complex, a buildup of acetyl-CoA from either the citric acid cycle or fatty acid oxidation signals that the cell is energetically satisfied and that the production of acetyl-CoA should be slowed or stopped. Pyruvate can then be used to form other products such as oxaloacetate for use in gluconeogenesis. Glycogenesis and Glycogenolysis. Glycogen, a branched polymer of glucose, represents a storage form of glucose. Glycogen synthesis and degradation occur primarily in liver and skeletal muscle, although other tissues store smaller quantities. Glycogen is stored in the cytoplasm as granules. Each granule has a central protein core with polyglucose chains radiating outward to form a sphere, as shown in figure 9.7. Glycogen granules composed entirely of linear chain chains have the highest density of glucose near the core. If the chains are branched, the glucose density is highest at the periphery of the granule, allowing more rapid release of glucose on demand. So linear chains have higher density at the core, branch chains have higher density in the periphery. Glycogen. Primarily in the liver and skeletal muscle. Glycogen stored in the liver is a source of glucose that is mobilized between meals to prevent low blood sugar, whereas muscle glycogen is stored as an energy reserve for muscle contraction. While our focus is on human metabolism, it is worth mentioning that plants also store excess glucose in long alpha-linked chains of glucose called starch, as seen in figure 9.8. And they have a picture of a potato. Great. Glycogenesis. Glycogenesis is the synthesis of glycogen granules. It begins with a core protein called glycogenin. As shown in figure 9.9, glucose addition to a granule begins with glucose 6-phosphate, which is converted to glucose 1-phosphate. This glucose 1-phosphate is then activated by coupling to a molecule of uridine diphosphate, UDP, which permits its integration into the glycogen chain by glycogen synthase. This activation occurs when glucose 1-phosphate interacts with uridine triphosphate, forming UDP glucose and pyrophosphate. So glycogenin is the core protein. Starts with G6P, which is converted to G1P, so G1P to UDP glucose, UDP glucose. UTP to PPI. All right. Glycogen synthase. Glycogen synthase is the rate-limiting enzyme of glycogen synthesis and forms the alpha-1,4 glycosidic bond found in the linear glucose chains of the granule. It is stimulated by glucose 6-phosphate and insulin. It is inhibited by epinephrine and glucagon through a protein kinase cascade that phosphorylates and inactivates the enzyme. Glycogen synthase is stimulated. The alpha-1,4 glycosidic bond. Is that important? Sure. Alpha-1,4 glycosidic bond. Stimulated by G6P and insulin. And it's inhibited by epinephrine and glucagon. All right. Branching enzyme. Glycosyl alpha-1,4 alpha-1,6 transferase. Branching enzyme is responsible for introducing alpha-1,6 linked branches into the granule as it grows. The process by which the branch is introduced is shown schematically in figure 910. Branching enzyme hydrolyzes one of the alpha-1,4 bonds to release a block of oligoglucose, a few glucose molecules bonded together in a chain, which is then moved and added in a slightly different location. Then it forms an alpha-1,6 bond to create a branch. Glycogen synthase makes a linear alpha-1,4 linked glucose chain. Branching enzyme hydrolyzes an alpha-1,4 bond. Branching enzyme then transfers the oligoglucose unit and attaches it with an alpha-1,6 bond to create a branch. And then glycogen synthase extends both branches. So branching enzyme is technically glycosyl alpha-1,4 to alpha-1,6 transferase. And it makes your alpha-1,4 to an alpha-1,6, as you might imagine. That's not even necessary, is it? Glycogenolysis. So this is glycogenesis. And now let's get into glycogenolysis. The rate-limiting enzyme of glycogenolysis. The process of breaking down glycogen is glycogen phosphorylase. In contrast to a hydrolase, a phosphorylase breaks bonds using an inorganic phosphate instead of water. The glucose 1-phosphate formed by glycogen phosphorylase is converted to glucose 6-phosphate by the same mutase used in glycogen synthesis, as shown in figure 9.9. Should I also write down that glycogen synthase is rate limiting? In glycogenolysis, we have glycogen phosphorylase. So glycogen phosphorylase takes glycogen to glucose 1P and then a mutase used in same mutase used in glycogen synthesis, and that mutase is I didn't write that down, did I? To G6P by a mutase and make this blue too. And then we have glycogen phosphorylase. Oh, that is the first step. Glycogen phosphorylase breaks alpha-1,4 glycosidic bonds, releasing glucose 1-phosphate from the periphery of the granule. It cannot break alpha-1,6 bonds and therefore stops when it nears the outermost branch points. Glycogen phosphorylase is activated by glucagon in the liver so that glucose can be provided for the rest of the body. In skeletal muscle, it is activated by AMP and epinephrine, which signal that the muscle is active and requires more glucose. It is inhibited by ATP can't break 1,6 linkages. Yeah, I can't break alpha 1,6. Only alpha 1,4. It's activated by glucagon in the liver. AMP and epinephrine in muscle. But it's inactive. It's inhibited by ATP. D-branching enzyme. Glucosyl alpha 1,4 to alpha 1,4 transferase and alpha 1,6 glucosidase. Branching enzyme is a two-enzyme complex that deconstructs the branches in glycogen that have been exposed by glycogen phosphorylase. The two-step process by which this occurs is diagrammed in figure 911. Debranching enzyme breaks an alpha 1,4 bond adjacent to the branching point and moves the small oligoglucose chain that is released to the exposed end of the other chain. Then it forms a new alpha 1,4 bond, hydrolyzes the alpha 1,6 bond, releasing the single residue at the branching point as free glucose. This represents the only free glucose produced directly in glycogenolysis, as opposed to the glucose produced from glucose 1-phosphate, which must be converted by a mutase to glucose 6-phosphate before it can be converted to glucose via the enzyme glucose 6-phosphatase. So as opposed to glycogen phosphorylase, the free glucose produced here does not need to be mutated. So glycogen phosphorylase releases glucose 1P from the periphery of the granule until it encounters the first branch. 2, debranching enzyme hydrolyzes the alpha 1,4 bond nearest the branch point. 3, branching enzyme transfers the oligoglucose unit to the end of another chain. And then it hydrolyzes the 1,6 bond, releasing the single glucose from the former branch. debranching enzyme. Glucosyl alpha 1,4 to alpha 1,4 transferase and alpha 1,6 glucosidase. So I'm just going to write that it cleaves 1 alpha 1,4 and alpha 1,6. Although it does it in separate steps, it can do both. All right, glycogen storage diseases. There are a number of genetic deficiencies that can impact the metabolism of glycogen. The clinical features of metabolic glycogen defect depend on a few important factors: which enzyme is affected, the degree to which that enzyme's activity is decreased, and which isoform of the enzyme is affected. Isoforms are slightly different versions of the same protein. In the case of glycogen enzymes, there are often different isoforms of the enzyme in the liver and muscle. These deficiencies are termed glycogen storage diseases because all are characterized by accumulation or lack of glycogen in one or more tissues. So let's just write down there exist glycogen storage diseases. What is the structure of glycogen? What types of glycosidic links exist in a glycogen granule? Well, the structure is branched. The linear portions are 1,4 linkages, and the branches come from 1,6, alpha 1,6 linkages. 2. What are the two main enzymes of glycogenesis, and what does each accomplish? Two main types. Oh, the two main enzymes of glycogenesis are glycogen synthase and branching enzyme. The glycogen synthase just makes the linear 1,4 linkages, while the branching enzyme transfers one of the 1,4s to a 1,6, making the branch. What are the two main enzymes in glycogenolysis, and what does each accomplish? In glycogenolysis, you have the glycogen phosphorylase, which is going to break up 1,4 bonds, and then the debranching enzyme will break up the 1,6 bond. Let us double check those answers. Glycogen is made up of a core protein with linear chains connected by 1,4 links and branches composed of 1,6 links. Glycogen synthase attaches the glucose molecule from UDP glucose to the growing glycogen chain, forming 1,4 links. Branching enzyme breaks the 1,4 link and makes the 1,6 link. Glycogen phosphorylase removes the glucose molecule from glycogen using a phosphate, breaking the 1,4 link, creating a glucose 1-phosphate. Debranching enzyme moves all of the glucose from a branch to a longer glycogen chain by breaking the 1,4 link, forming a new 1,4 link to the longer chain. And then the branch that's left behind is removed by breaking the 1,6 link. to make a free glucose. Great. Gluconeogenesis. And then the pentose phosphate pathway. I'm going to go to the bathroom real quick, take a quick break, and then we'll continue. All right, gluconeogenesis. The liver maintains glucose levels in blood during fasting through either glycogenolysis or gluconeogenesis. The kidney can also carry out gluconeogenesis, although its contribution is much smaller. These pathways are promoted by glucagon and epinephrine, which act to raise blood sugar levels, and are inhibited by insulin, which act to lower blood sugar levels. During fasting, glycogen reverses During fasting, Glycogen reserves drop dramatically in the first 12 hours, during which time gluconeogenesis increases. After 24 hours, it represents the sole source of glucose. Important substrates for gluconeogenesis are glycerol 3-phosphate from stored fats or triacylglycerols in adipose tissue, lactate from anaerobic glycolysis, and glucogenic amino acids from muscle proteins. Gluconeogenesis. Liver and kidneys. Promoted by glucagon and epinephrine. Inhibited by insulin. So it's important during fasting. Important for fasting. Substrates include G3P, lactate, and amino acids. G3P, lactate, and glucogenic amino acids. All right, the last item of this list merits some explaining. Amino acids can be subclassified as glucogenic, ketogenic, or both. Glucogenic amino acids, all except leucine and lysine, can be converted into intermediates that feed into gluconeogenesis, while ketogenic amino acids can be converted into ketone bodies, which can be used as an alternative fuel, particularly during periods of prolonged starvation. See Chapter 11 for more information on amino acid and protein metabolism. So gluconeogenesis, an alternative to gluconeogenesis would be ketosis. So glucogenic versus ketogenic. And while gluconeogenesis is associated with fasting 12-24 hours, ketosis is associated with prolonged starvation. Prolonged starvation. Dietary fructose and galactose can also be converted to glucose in the liver, as described earlier in this chapter. In humans, while glucose is converted into acetyl-CoA through glycolysis and pyruvate dehydrogenase, it is not possible to convert acetyl-CoA back to glucose. Because most fatty acids are metabolized solely to acetyl-CoA, they are not a major source of glucose either. One minor exception is fatty acids with an odd number of carbon atoms. For example, fatty acid tails containing 17 carbons, which yield a small amount of propionyl-CoA, which is glucogenic. So that's just an odd little fact, that odd fatty acids make a little bit of propionyl-CoA, which is glucogenic. The pathway of gluconeogenesis is diagrammed in figure 912. Each of the important gluconeogenic intermediates, lactate, alanine, and glycerol 3-phosphate, have enzymes that convert them into glycolytic intermediates. Alanine, we learned about, is alanine the glucogenic amino acid? Alanine into pyruvate. All right. We have fatty acids on here. So the glycerol 3-phosphate, that's the glycogen. Okay, acetyl-CoA from fatty acids. This is your fatty acid part. They said the important parts are lactate, alanine, and G3P. Alanine's got alanine aminotransferase. Lactate, where is the lactate part? Lactate dehydrogenase. Great. Should we note some of these? Are we going to talk about these specifically? Important enzymes. We will talk about these. So while glycolysis is glucose down to pyruvate, this is pyruvate back up to glucose. What were the other ones that we went over? We went over glycogen. That's a little glycogenesis and glycogenolysis. This is gluconeogenesis, all the way back up to glucose, not some other storage form. And so like glucokinase would be like your glycolysis form. Glucose 6-phosphatase is your gluconeogenesis counterpart. PFK is an irreversible enzyme. So in gluconeogenesis, you have a counterpart, F16-biphosphatase. Great. Should we write some of these down? That lactate dehydrogenase, alanine aminotransferase, and glycerol 3P. Dehydrogenase. Lactate is converted to pyruvate by lactate dehydrogenase. Alanine is converted to pyruvate by alanine aminotransferase. And glycerol 3-phosphate is converted to dihydroxyacetone phosphate, DHAP, by glycerol 3-phosphate dehydrogenase. Important enzymes of gluconeogenesis. Most steps in gluconeogenesis represent a reversal of glycolysis and have thus been omitted from the diagram. However, the 4 important enzymes to know are those required to catalyze reactions that circumvent the irreversible steps of glycolysis in the liver. Those catalyzed by glucokinase, phosphofructokinase 1, and pyruvate kinase. Where was the pyruvate kinase one? Pyruvate kinase. So we have to take pyruvate through OAA back to PEP. All right, let's read about that. Pyruvate carboxylase. So these are going to be for intermediates specifically. Pyruvate carboxylase, and these are going to be reversals of irreversible enzymes in glycolysis. Pyruvate carboxylase is a mitochondrial enzyme that is activated by acetyl-CoA from beta oxidation. The product, oxaloacetate, OAA, is a citric acid cycle intermediate and cannot leave the mitochondria. Rather, it is reduced to malate, which can leave the mitochondria via the malate aspartate shuttle, which is described in Chapter 10. Once in the cytoplasm, malate is oxidized to OAA. The fact that acetyl-CoA activates pyruvate carboxylase is an important point. Acetyl-CoA inhibits pyruvate dehydrogenase because a high level of acetyl-CoA implies that the cell is energetically satisfied and need not run the citric acid cycle in the forward direction. In other words, the cell should stop burning glucose. Rather, pyruvate will be shunted through pyruvate carboxylase to help generate additional glucose through gluconeogenesis. Note that the source of acetyl-CoA is not from glycolysis and pyruvate dehydrogenase in this case, but from fatty acids. Thus, to produce glucose in the liver during gluconeogenesis, fatty acids must be burned to provide this energy, stop the forward flow of the citric acid cycle, and produce massive amounts of OAA that can eventually lead to glucose production for the rest of the body. All right. So it's in the mitochondria. Activated by acetyl-CoA. From beta oxidation, I guess. The product is OAA. So it takes, I guess, pyruvate to oxaloacetate, which is abbreviated OAA, and then to malate, aspartate shuttle, and then back into oxaloacetate. So that's your little shuttle, the oxaloacetate. Back to malate. So they're saying that this pyruvate, we're not actually making pyruvate, we're making OAA. They're saying that this comes from fat. This acetyl-CoA comes from fatty acids. So they don't show the acetyl-CoA here. Beta oxidation is fatty acid degradation, I guess. Fatty acids. All right. Phosphoenol pyruvate carboxykinase. Tca. Phosphoenol pyruvate carboxykinase in the cytoplasm is induced by glucagon and cortisol, which generally act to raise blood sugar levels. It converts OAA to PEP, phosphoenol pyruvate, in a reaction that requires GTP. PEP continues in the pathway to fructose 1,6-biphosphate. Thus, the combination of pyruvate carboxylase and Tca are used to circumvent the action of pyruvate kinase by converting pyruvate back into PEP. Phosphoenol pyruvate carboxykinase, or Tca, induced by glucagon and cortisol, converts OAA to PEP, and requires GTP. And then it says the next step after that is it continues to fructose 1,6-biphosphate by a bunch of reactions, so they just skip over those. All right. Fructose 1,6-biphosphatase. The next irreversible step. So between the cytoplasm and the mitochondria, you have this malate aspartate shuttle. So I imagine gluconeogenesis doesn't work that well when that shuttle isn't working well. So if you have mitochondrial, I mean, there's so many different kinds of mitochondrial issues, I guess. Tca. Fructose 1,6. Biphosphatase. Fructose 1,6-biphosphatase is the site in the cytoplasm, is a key control point of gluconeogenesis and represents the rate-limiting step of the process. It reverses the action of phosphofructokinase 1, the rate-limiting step of glycolysis, by removing phosphate from fructose 1,6-biphosphate to produce fructose 6-phosphate. A common pattern to note is that phosphatases oppose kinases. There you go. Fructose 1,6-biphosphatase is activated by ATP and inhibited by AMP and fructose 2,6-biphosphate. This should make sense. High levels of ATP imply that a cell is energetically satisfied, enough to produce glucose for the rest of the body, whereas high levels of AMP imply that a cell needs energy and cannot afford to produce energy for the rest of the body before satisfying its own requirements. Fructose 2,6-biphosphate is sometimes thought of as a marker for satisfactory energy levels in liver cells. It helps these cells override the inhibition of PFK that occurs when high levels of acetyl-CoA are formed, signaling to the liver cell that it should shift its function from burning to storing fuel. F26-biphosphate, produced by PFK2, controls both gluconeogenesis and glycolysis in the liver. Recall from the earlier discussion of this enzyme in figure 9.2 that PFK2 is activated by insulin and inhibited by glucagon. Thus, glucagon will lower F26-biphosphate and stimulate gluconeogenesis, whereas insulin will increase F26BP and inhibit gluconeogenesis. All right. Biphosphatase takes F16BP to F6P. Note that phosphatases oppose kinases. It's activated by ATP, inhibited by AMP, so F26BP, the alternate fructose biphosphate, is a marker for energy in the liver. It overrides the inhibition of PFK. It's produced by PFK2. So it overrides inhibition of PFK1 by acetyl-CoA. And it should store energy. So PFK2 through the mechanism of F26BP controls both gluconeogenesis and glycolysis. PFK2 is activated by insulin and inhibited by glucagon. Thus, glucagon will lower F26BP and stimulate gluconeogenesis, whereas insulin will increase F26BP and inhibit gluconeogenesis. So PFK2 is upregulated by insulin. So increase in insulin means more F26BP. And less gluconeogenesis. And glucagon or inhibited by glucagon, which causes less F26BP and stimulate gluconeogenesis. All right. All right. Glucose 6-phosphatase is found only in the lumen of the endoplasmic reticulum in liver cells. Glucose 6-phosphate is transported into the ER, and free glucose is transported back into the cytoplasm, from where it can diffuse out of the cell using glut transporters. The absence of glucose 6-phosphatase in skeletal muscle means that muscle glycogen cannot serve as a source of blood glucose and rather is for use only within the muscle. Glucose 6-phosphatase is used to circumvent glucokinase and hexokinase, which convert glucose to glucose 6-phosphate. Glucose 6-phosphate in the liver and it circumvents glucokinase or hexokinase. Although alanine is the major glucogenic amino acid, almost all amino acids are also glucogenic. Most of these are converted by individual pathways to citric acid cycle intermediates, then to malate, following the same path from there to glucose. Did I write down alanine before? Alanine aminotransferase. Citric acid cycle intermediates are not acetyl-CoA, that's beta oxidation. Alanine aminotransferase. This doesn't have the citric acid cycle on it, so we don't really even know much about that, do we? So let's just write down the related to citric acid cycle, malate. It is important to note that glucose produced by hepatic gluconeogenesis does not represent an energy source for the liver. Gluconeogenesis requires expenditure of ATP that is provided by beta oxidation of fatty acids. Therefore, as mentioned above, hepatic gluconeogenesis is always dependent on beta oxidation of fatty acids in the liver. During periods of low blood sugar, adipose tissue releases these fats by breaking down triacylglycerols to glycerol, which can also be converted to the gluconeogenic intermediate DHAP and free fatty acids. So the ATP comes from beta oxidation of fatty acids. All of the acetyl-CoA from fatty acids cannot be converted into glucose. It can be converted into ketone bodies as an alternative fuel for cells, including the brain. Extended periods of low blood sugar are thus usually accompanied by high levels of ketones in the blood. Ketone bodies can be thought of as transportable forms of acetyl-CoA that is primarily utilized in periods of extended starvation. So although acetyl-CoA can't be turned into glucose, it can be transported as ketone bodies. And then back into acetyl-CoA. For transportation of acetyl-CoA. All right, under what physiological conditions should the body carry out gluconeogenesis? When there's sufficient ATP, when there's sufficient sufficiently low AMP, when there's glucagon and not insulin. Oh no, so you do gluconeogenesis when there isn't sufficient ATP. It's a marker for energy in the liver. Sorry. Yeah, under what physiological conditions should the body carry out gluconeogenesis? I guess it's regulated by insulin. When there's glucagon and epinephrine, but not when there's insulin. What are the 4 enzymes unique to gluconeogenesis, and which irreversible glycolytic enzymes do they replace? Starting with Pyruvate Carboxylase. It replaces PEP decarboxylase. Pyruvate Kinase, Tca. Oh, so I guess Pyruvate Carboxylase and Tca function together to counteract Pyruvate Kinase. It's a two-step. We have fructose 1,6 biphosphatase is going to reverse the action of PFK1. We have glucose 6-phosphate phosphatase, which is going to reverse the activity of glucokinase. And that's all we talked about. Why are there 4 of them? Gluconeogenesis. We have Pyruvate Carboxylase, Tca. Oh, okay. So Tca and Pyruvate Carboxylase reverse the same enzyme together. But you're right, there's 2 enzymes. How does acetyl-CoA shift the metabolism of pyruvate? Acetyl-CoA let's see. Let's take a look at this again. Pyruvate, acetyl-CoA inhibits PDH and also inhibits pyruvate. Oh, it encourages pyruvate carboxylase. So the presence of acetyl-CoA promotes gluconeogenesis. Yeah, that'll do. Let's see what they say. (9.6) Gluconeogenesis occurs when an individual has been fasting for greater than 12 hours. To carry out gluconeogenesis, hepatic and renal cells must have enough energy to drive the process of glucose creation, which requires sufficient fat stores to undergo beta oxidation. All right. 2. Pyruvate carboxylase and Tca are both replacing pyruvate kinase. F16 biphosphate is replacing PFK, and G6P phosphatase is going to be replacing glucokinase or hexokinase. And 3, acetyl-CoA inhibits pyruvate dehydrogenase complex while activating pyruvate carboxylase. The net effect is to shift from burning pyruvate in the citric acid cycle to creating new glucose molecules for the rest of the body. The acetyl-CoA for this regulation comes predominantly from beta oxidation, not glycolysis. All right. (9.7) The pentose phosphate pathway. The pentose phosphate pathway, PPP, also known as the hexose monophosphate shunt, HMP shunt, occurs in the cytoplasm of all cells where it serves 2 major functions: production of NADPH and serving as a source of ribose 5-phosphate 4 nucleotide synthesis. Now, sorry to stop so quick, but gluconeogenesis is still mostly in the cytoplasm as well. Only part of it's in the mitochondria, I guess. All right. So what is in the mitochondria? Beta oxidation. Oxidative. That's in the next chapter, right. So these two major functions is a production of NADPH and serving as a source of ribose 5-phosphate for nucleotide synthesis. Produces NADPH and ribose 5-phosphate. For nucleotide synthesis. An abbreviated diagram of the pathway is shown in figure 9.13. So let's write down that these go to nucleotides. An abbreviated diagram of the pathway. The first part of the PPP begins with glucose 6-phosphate, ends with ribose 5-phosphate, and is irreversible. This part produces NADPH and involves the important rate-limiting enzyme glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase. G6PD. G6PD is induced by insulin because the abundance of sugar entering the cell under insulin stimulation will be shunted into both fuel utilization pathways, glycolysis and aerobic respiration, as well as fuel storage pathways, fatty acid synthesis, glycogenesis, and the PPP. The shunt is also inhibited by its product, NADPH, and is activated by one of its reactants, NADP+. So it's induced by insulin. Induced by insulin and NADP+, and inhibited by NADPH. The second part of the pathway, beginning with ribulose 5. Oh, this is ribulose, not ribose. ribulose. Second part of the pathway begins with ribulose 5-phosphate, represents a series of reversible reactions that produce an equilibrated pool of sugars for biosynthesis, including ribose 5-phosphate for nucleotide synthesis. Because fructose 6-phosphate and glyceraldehyde 3-phosphate are among the sugars produced, intermediates can feed back into glycolysis. Conversely, pentoses can be made from glycolytic intermediates without going through the G6PD reaction. These interconversions are primarily accomplished by the enzymes transketolase and transaldolase. So the first one is irreversible, or G6PD. But the rest of these are going to be reversible. I'm going to make a bunch of different sugars. So let's just write down transketolase and transaldolase. Functions of NADPH. While their names appear similar, NADPH and NADH are not the same thing. In the cell, NAD+ acts as a high-energy electron acceptor from a number of biochemical reactions. It thus can be thought of as a potent oxidizing agent because it helps another molecule be oxidized, and thus is reduced itself during the process. The NADH produced from this reduction of NAD+ can then feed into the electron transport chain to indirectly produce ATP. So let's talk about NADPH and NAD plus. Is an oxidizing agent. Conversely, NADPH primarily acts as an electron donor in a number of biochemical reactions. It thus can be thought of as a potent reducing agent because it helps other molecules be reduced and thus is oxidized itself during the process. Cells require NADPH for a variety of functions, including biosynthesis, mainly of fatty acids and cholesterol, assisting in cellular bleach production in certain white blood cells, thereby contributing to bactericidal activity. NADPH is a reducing agent. Cellular bleach. White blood cells actually have bleach in them? Interesting. NADPH also is used in the maintenance of a supply of reduced glutathione or glutathione to protect against reactive oxygen species, acting as the body's natural antioxidant. This last function is important in protecting cells from free radical oxidative damage caused by peroxides. Hydrogen peroxide, H2O2, is produced as a byproduct in aerobic metabolism and can break apart to form hydroxide radicals, OH radicals. Free radicals can attack lipids, including those in the phospholipids of the membrane. When oxidized, these lipids lose their function and can weaken the membrane, causing cell lysis. This is especially true in red blood cells, which contain high levels of oxygen, which, when oxidized by other free radicals, become the superoxide radical O2 radical. Free radicals can also damage DNA, potentially causing cancer. Glutathione is a reducing agent that can help reverse radical formation before damage is done to the cell. So NADPH contributes to glutathione, which is an antioxidant. All right. What are two major metabolic products of the pentose phosphate pathway? Metabolic products, NADPH, and ribose for nucleotide synthesis, ribose 5-phosphate. What are three primary functions of NADPH? It's used in synthesis of fatty acids and cholesterol. It's used to make bactericidal activity, and it's used to make the antioxidant glutathione. All right. Can you check the answers? Pretty confident about those. Two major metabolic products are ribose 5-phosphate and NADPH. NADPH is involved in lipid synthesis, bactericidal bleach formation in white blood cells, and to maintain glutathione stores to protect against reactive oxygen species. Great. All right, discrete practice questions. Number 1, a man collapses while running a marathon and is taken to the emergency room. His blood is found to be somewhat acidic, and further tests show increased lactate dehydrogenase activity. This enzyme is involved in which of the following pathways? Anaerobic glycolysis, beta oxidation of fatty acids, citric acid cycle, or pentose phosphate pathway. I think that's the anaerobic parts of glycolysis, right? Let's look at our glycolysis map. This is going to be useful. Lactate dehydrogenase, lactate. What was the question? Okay, well, it's anaerobic glycolysis. Lactate dehydrogenase. It's not. Yeah, 1A. 2. Which of the following organs does not require a constant supply of glucose from the blood for energy during a fast? Red blood cells, brain, pancreas, liver. I bet liver is the one that doesn't. Three, when insulin is released, it acts to increase the absorption of glucose into skeletal muscle predominantly through which of the following transporters? It's either 2 or 4. Let's double check. I think 4 is the one in the liver. 2 is the one elsewhere. So I'd say 2. GLUT2. No, I was wrong. Switch that. GLUT4 is in adipose and muscle. GLUT2 is in the liver. So GLUT2 is in the liver. GLUT4 is everywhere. D, GLUT4. 3, 4. After an overnight fast, which of the following enzymes would be expected to have little, if any, physiological activity? Malate dehydrogenase, glucokinase, alpha-ketoglutarate dehydrogenase, or phosphofructokinase 1. After fasting for 12 hours, you would be doing gluconeogenesis. I bet you'd be doing glycogenolysis. Or would you be doing gluconeogenesis? During fasting, glycogen reserves drop dramatically in the first 12 hours, during which time gluconeogenesis increases. So which would have little activity would be ones involved in glycolysis. Malate dehydrogenase, that sounds like it's part of this. So that would be part of gluconeogenesis. Glucokinase would be glycolysis. Alpha-ketoglutarate dehydrogenase. Do I remember seeing that? Alpha-ketoglutarate. So it's a ketone. So that's probably in ketosis. So that's maybe not after just an overnight fast. Phosphofructokinase is also with glucokinase in glycolysis. So I would expect, I think malate dehydrogenase, gluconeogenesis. I'm going to go with A. 5. The diagram below shows the effects of arsenic on the metabolism of glyceraldehyde 3-phosphate. So, my bad. Malate dehydrogenase would be active. Glucokinase, phosphofructokinase, or ketoglutarate dehydrogenase. So the least activity would be the keto. You haven't had enough for keto yet. My bad. 4 should be C. Malate dehydrogenase, you would have the most of. Glucokinase and phosphofructokinase, I mean, you also still have to have some activity of that in some cells. The liver would have the malate dehydrogenase. The brain would have glucokinase and phosphofructokinase. All right, 5. The diagram below shows the effect of arsenic on the metabolism of glyceraldehyde 3-phosphate. As a result, in the presence of arsenic, how many molecules of ATP would be created directly from the conversion of 2 glucose molecules to 4 pyruvate molecules? So arsenic takes G3P directly to 3-phosphoglycerate without making ATP. As a result, in the presence of arsenic, how many molecules of ATP would be created directly from the conversion of 2 glucose molecules to 4 pyruvate molecules? So usually it makes 2, right? This one doesn't have our ATP. Does the first chart have ATP? This one does have ATP. We got ATP up here. We're using ATP up here. And then we're getting ATP back here. ATP to ADP, ATP to ADP. NAD plus to NADH. Here we make an ATP, here we make an ATP. So it seems like you're losing. It doesn't seem like you're making any. So one glucose molecule it doesn't look like you're making any because you're losing 2 and then gaining 2. Better plug this in. I'm going to say 2 because earlier we talked about how you get 2 ATP out of glycolysis. And if this is basically shutting one of those down, then so G3P to phosphoglycerate. G3P to 3-phosphoglycerate. So ignoring these, that 1, 2 that you would have made I want to say none, because that's probably why arsenic is bad, right? Because it shuts down metabolism. But it looks like it'd be maybe 2. But really, I mean, we're not making any ATP here. So if you shut this down, you'd be making negative ATP. -2. I'd like to say -2, but that's not an option. So I'm going to go 0. A. 6. When fatty acid beta oxidation predominates in the liver, mitochondrial pyruvate is most likely to be carboxylated to phosphoenol pyruvate for entry into gluconeogenesis, oxidatively decarboxylated to acetyl-CoA for oxidation in the citric acid cycle, carboxylated to oxaloacetate for entry into gluconeogenesis, reduced to lactate in the process of fermentation. Mitochondrial pyruvate could be carboxylated to oxaloacetate. That sounds right. So when you have a lot of acetyl-CoA from beta oxidation, it inhibits pyruvate to acetyl-CoA. It promotes pyruvate to oxaloacetate. So oxaloacetate sounds the best to me. Let's go with 6. 7 is going to be a biopsy is done on a child with an enlarged liver and shows accumulation of glycogen granules with single glucose residues remaining at the branch points near the periphery of the granule. The most likely genetic defect is in the gene encoding. So if you're not taking the single glucose residues at the branches, that means that your 1,6 isn't working. Your part of debranching enzyme, but the 1,6 glucosidase or glucosidase. I think it's going to be D. I'm going to scroll up here just to double check. Alpha 1,6 glucosidase. D. 8. An investigator is measuring the activity of various enzymes involved in reactions of intermediary metabolism. One of the enzymes has greatly decreased activity compared to reference values. The buffer of the assay contains citrate. Which of the following enzymes will most likely be directly affected by the use of citrate? So one of these was directly inhibited by citrate, right? Which was it? PFK is regulated by citrate. Yeah, phosphofructokinase 1. Let's go with that. C. After a brief period of intense exercise, the activity of muscle pyruvate dehydrogenase is greatly increased. This increased activity is most likely due to pyruvate dehydrogenase. Is that the last step of... That's not the last step of glycolysis. Pyruvate kinase is the last step of glycolysis. Pyruvate dehydrogenase is to get pyruvate back into the cell. Pyruvate dehydrogenase takes pyruvate to acetyl-CoA. Never mind. These are the reverse, since this is an irreversible reaction. Pyruvate dehydrogenase is greatly increased. It's probably not because of acetyl-CoA, because it's inhibited by acetyl-CoA. Could just be pyruvate, decreased ADP, What does pyruvate dehydrogenase actually do? Pyruvate dehydrogenase is pyruvate to acetyl-CoA in the mitochondria. So it wouldn't be increased acetyl-CoA, it could be increased pyruvate, or it could be an increased NADH to NAD plus ratio. Now NAD plus is the cofactor, so a greater NADH to NAD plus ratio also would not help. Decreased ADP, increased ADP would also do it, not decreased ADP. So increased pyruvate concentration, let's go with D. So NAD plus and NADH. NAD plus is the oxidizing agent. So if there's more NADH to NAD plus, then we're like out of oxidizer. And that is something that you'll use in aerobic respiration. So they didn't say after eating. They said after intense exercise. So I don't think it's pyruvate concentration. It's got to be NADH to NAD plus ratio. It has to do with aerobic, oxidative phosphorylation. So we're going to go with C. 9 is C. 10, after a large, well-balanced meal, all of the following substances would be expected to be elevated except fatty acids, insulin, glucose, glucagon. So after eating, you'll have high blood sugar, and so you'll have insulin to get that sugar into the cells. Glucagon is the one that I think won't be in high supply, right? Because glucagon is what would, for example, stimulate gluconeogenesis, which shouldn't be necessary in this case. All right, 11. A man is given antibiotics to treat a urinary tract infection and develops an episode of red blood cell lysis. Further studies show weakness of the plasma membrane and Heinz bodies, collections of oxidized hemoglobin. Which of the following enzymes is most likely defective in this patient? So they're just talking about antioxidants. So it's the pentose phosphate pathway. Which part of it? G6 phosphate dehydrogenase. That's part of the pentose phosphate pathway. The others are all part of glycolysis. So let's go with B. 12. The unique enzymes of gluconeogenesis are used to circumvent specific irreversible steps of glycolysis. Which of the following correctly pairs an enzyme from glycolysis with its corresponding enzyme in gluconeogenesis? PFK and fructose 1,6 biphosphatase. That sounds right. Could it be? They have a great graph of it. Let's peek at that. PFK, fructose 1,6 biphosphatase. Did I write these down? Didn't actually write down what these counter, but I'm going to do it right now. These two together counteract pyruvate kinase, anti-pyruvate kinase. Fructose 1,6 is anti-PFK. And G6P phosphatase is anti-glucokinase. Anti-glucokinase. 12. Phosphofructokinase and fructose 1,6, that sounds good. Pyruvate dehydrogenase and pyruvate carboxylase. And phosphoenol pyruvate carboxykinase. I don't know about that. Hexokinase and glucokinase, those are not opposites, they're just in different cells. Pyruvate kinase and glucose-6-phosphatase. No. The opposite of pyruvate kinase was phosphoenol pyruvate carboxylase. Phosphoenol pyruvate carboxylase is the opposite of pyruvate kinase. So A is the only one that's right. A. 13 is overnight fast. Which of the following processes would be expected to occur at an elevated rate compared with the well-fed state? Glycolysis, glycogenolysis, glycogenesis, or glycerol synthesis? Glycogenolysis. 13B. Glycogenesis is making fat, or glycogen. And glycerol synthesis, isn't that just a part of glycolysis? The glycogen, or the glycerol synthesis, that's in the pathway. It's an alternative pathway to glycolysis. So yeah, glycogenolysis, the lysis of glycogen. 14. Which of the following are functions of NADPH in the cell? NADPH. Antimicrobial resistance, via bacterial destruction by bleach within lysosomes. Antimicrobial resistance or microbial resistance? Sure. 2. Involvement in the production of the precursors to steroid hormones. 3. Functional carriage of energy across organelle membranes for use within the mitochondria. 1 is the only one that looks right to me. I don't know if it makes steroid hormones. I think it makes some things. Let's see. NADH, biosynthesis mainly of fatty acids and cholesterol. Oh yeah, cholesterol. So that is a steroid hormone, right? It's in fatty acids, cholesterol, bleach, and antioxidants. So it is 1, 2, 3 functional carriage of energy across organelle membranes for use within the mitochondria. Is that one of the things that it does? Here's a mitochondria. NAD plus is the oxidizing agent. NADPH is the reducing agent. I'm going to go with 1 and 2 only, but not 3. (15) Each of the following catalyzes a rate-limiting step of a carbohydrate metabolism pathway except hexokinase, glycogen synthase, glucose 6-phosphate dehydrogenase, or fructose 1,6-biphosphatase. Fructose 1,6-biphosphatase is the opposite of PFK, so that is a rate-limiting step. I think hexokinase is also irreversible. Glycogen synthase. Glycogenesis. Glycogen synthase is a rate-limiting step. G6-phosphate dehydrogenase. G6PD. It is also irreversible. Fructose 1,6-biphosphatase. I already thought that was. Glucogenesis, fructose 1,6-biphosphate, biphosphatase. Hexokinase, I guess, isn't the rate-limiting step. It's irreversible, but PFK is considered the rate-limiting step, right? PFK is rate limiting. Hexokinase is not. Glycogen synthases, G6PD. Let's see. G6PD. Is G6PD the rate-limiting step of the pentose phosphate pathway? Let's double check. Pentose phosphate pathway. This part produces and involves an important rate-limiting enzyme. G6PD is rate-limiting. So yeah, hexokinase has got to be the answer. Because this one is, this one is, this one is. All right. Let's see how we did. Number 1, A. 2, D. 3 is D. 4 is B, not C. After an overnight fast, the liver is producing glucose and glucokinase activity would be insignificant. Glucokinase is used to trap extra glucose in liver cells as part of a storage mechanism. With low blood glucose, liver cells would be creating new glucose, not storing it. It is also in the pancreas where it serves as a glucose sensor. If glucose levels are low, it has little activity in this tissue as well. Malate dehydrogenase, choice A, and alpha-ketoglutarate dehydrogenase, choice C, are citric acid cycle enzymes. PFK, choice D, is a glycolytic enzyme. Other enzymes used in glycolysis, the citric acid cycle or gluconeogenesis, such as PFK1, would be expected to maintain normal activity overnight fast, using glucose derived from glycogen or gluconeogenesis rather than orally ingested glucose. So they said glucokinase would have little activity. Because glucokinase is in the liver, not hexokinase, which is in all cells. So the liver wouldn't be taking up sugar at this point. It would probably be releasing it for the rest of the body. Hexokinase you would expect to be normal. Glucokinase you would not. All right. Alpha-ketoglutarate and dehydrogenase and malate dehydrogenase, they said, were both part of the citric acid cycle. PFK, on the other hand, what did I say? I said C, alpha-ketoglutarate dehydrogenase. So, after an overnight fast, would you not expect to see citric acid cycle enzymes? All right. Well, number 5 is A, 6 is C, 7 is D, 8 is C, 9 is D, not C. In most biochemical pathways, only a few enzymatic reactions are under regulatory control. These often occur either at the beginning of pathways or at pathway branch points. The Pyruvate Dehydrogenase, PDH complex controls the link between glycolysis and the citric acid cycle and decarboxylates pyruvate, the end product of glycolysis. With production of NADH and acetyl-CoA, the substrate for the citric acid cycle. After intense exercise, one would expect PDH to be highly active to generate ATP. ADP levels, choice A, should be high because ATP was just burned by the muscle. Acetyl-CoA, choice B, is an inhibitor of PDH, causing a shift of pyruvate into the gluconeogenic pathway. A high NADH-NAD plus ratio, choice C, would imply that the cell is already energetically satisfied and not in need of energy, which would not be expected in intensely exercising muscle. So, 9. I thought it would be C, but NADH does mean there's a lot of energy. I thought a lot of NAD plus would mean a lot of energy. They're saying a lot of NADH means a lot of energy. Acetyl-CoA was the inhibitor. Decreased ADP after intense exercise wouldn't expect PDH to be highly active to generate ATP. ADP levels, choice A, should be high because ATP was just burned by the muscle. I also thought there would be ADP, so decreased ADP doesn't sound right. They said D, increased pyruvate concentration. Why is that? The PDH complex controls the link between glycolysis and the citric acid cycle and decarboxylates pyruvate, the end product of glycolysis, with production of NADH and acetyl-CoA, the substrate for the citric acid cycle. After intense exercise, one would expect PDH to be highly active to generate ATP. Because you'd expect PDH breaks pyruvate into acetyl-CoA. And you would expect to see it. So you'd also have pyruvate concentration because of pyruvate dehydrogenase. But if you'd expect pyruvate dehydrogenase to be active, then wouldn't you expect a lot of acetyl-CoA, not a lot of pyruvate? So to generate ATP, you'd have high PDH levels. and it controls the link between glycolysis and the citric acid cycle. Is TCA also the citric acid cycle? What is the TCA cycle? It is a citric acid cycle. It's the tricarboxylic acid cycle. That's why it's TCA. So I guess they're saying that if this is active, so because this is going to be active, you're going to pull a lot of pyruvate into the mitochondria. So you will have a lot of pyruvate. You're going to be demanding a lot of pyruvate. Okay, that's 9. "10 is D, 11 is B, 12 is A, 13 is B, 14 is B, and 15 is A." That is it for Carbohydrate Metabolism 1. Next time, we'll get into Chapter 10, Carbohydrate Metabolism 2, Aerobic Respiration.